I'm Michael Garton. I'm a professor in biomedical engineering at the University of Toronto. My laboratory specializes in synthetic biology, particularly applying artificial intelligence and synthetic biology to mammalian engineering. So my lab is around 15-20 people, around a quarter of whom are working on developing artificial intelligences that are used to design biology. In particular, specifically biological sequences, so DNA sequences and protein sequences. And this is all toward integrating these sequences with human cells. We either put these into stem cells and differentiate them into different tissues, or we put them into modified viruses for delivery to humans. Yeah, so I started life as a chemistry undergrad. During my undergrad, I was also intending to be a professional climber. I'd got to a sort of semi-professional level in rock climbing. And then toward the end of my undergrad, I was trying to do the first solo ascent of a cliff called the Troll Wall in Norway, which is among climbing circles known to be one of, if not the most dangerous mountain wall in the world. It's the highest in Europe. And as you might expect, trying something like that, had an accident and ended up paralyzed from the neck down. So that changed my career a little bit, just a tiny bit. And I decided to go into computational chemistry. And from there, I did a master's in medicinal computational chemistry, and then went on to do a PhD. When I went to do a PhD, I decided to sort of swerve a little bit away from computational chemistry into computational biology. And so I studied telomeres and DNA structure at telomeres using computational simulation techniques. That was in Nottingham in the UK. I then came to Canada and worked as a postdoc at Sick Kids and in the University of Toronto. And then I wanted to push my career more toward making things that are going to impact the real world rather than just kind of studying things and working on theory. And so I branched more into protein design during my postdoc and started to design proteins that could be used as drugs. I kind of had the same feeling when I got a faculty position for my own lab that I wanted to push even more in the direction of making real translatable translational entities that will go out and be used in the real world and have real impact for regular everyday people. And so for my own lab, I decided to move even further from protein design to whole cell design, so human cell design. And so my lab is essentially synthetic biology of human cells and integrating synthetic biology with human cells. And so the idea is that we're building cells that can, for example, sense disease and then produce a therapeutic response. And then we can make those cells into implants or grafts that will then be implanted into someone for the rest of their life. That's the idea. And we'll kind of monitor and handle, therapeutically intervene with that disease in tune with the ebb and flow of the disease throughout that person's life. From there, we also moved into an area called gene therapy. And what gene therapy is rather than engineering the cells themselves, so for example, something we do is engineer stem cells, turn them into muscle, and then implant the muscle. So rather than doing that, we try to engineer the cells in the humans themselves. And so the idea is that we take common viruses and we take out the internal genome of the virus, so therefore it can't replicate, so it's a non-replicating virus, and we put a human gene into the cell, or a human synthetic circuit, and then we deliver that by just injecting the viruses, which then get delivered to hopefully the target tissue that you want. An example might be muscle. So for people who have muscular dystrophy, there's a gene that is defective or not made at all, and the idea is that if you can simply deliver a correct, non-deficient copy of that gene to the muscle cells, that you will restore the function of those muscles and permanently correct the disease. That's the idea. One of the projects we're working on is to make viruses, specifically viruses that can carry the DMD gene to human muscle. And so the lab is split between gene therapy, cell therapy, and then we have a computational team that do a lot of the design work by designing artificial intelligences that can design some of these biological sequences that we need to solve these problems. So there's definitely a lot of interest in synthetic biology across Canada. I think that the IGEM competition is a good reflection of that, which is undergrad student-led teams that then compete in an international competition. I mentored a team last year that had 90 students, and it was fantastic to work with them and see this sort of very. I wasn't leading it in any means. It was entirely student-led, and it was great to see lots of different students from lots of different backgrounds bringing their expertise to bear on this project, from kind of business people and people in law, all the way through to the sort of biological and biotechnology people. The vision really is to be able to deliver synthetic gene circuits with synthetic components that provide an augment human cells, like you would add an app to an iPhone. You add a synthetic circuit to a human cell, and it can now correct your blood clotting disorder or your muscular disease. But the main barriers and the things that we're working on are one, when you put anything into a cell that isn't there originally, anything synthetic, anything different to what humans normally have, cells recognize that pretty fast. And so one challenge is getting it past the immune system without the immune system noticing that this thing is in there. So that's one thing we're working on. Another thing that we're working on is something called epigenetic silencing. Another thing that happens when you put something synthetic into a human cell, the cell tries to shut down that genetic material. It recognizes it as non-self, not human, or even if it is, it can recognize it as a different human, and it tries to just close down and shut down that DNA so it can no longer express. And so one of the things that we're working on is designing it such that it cannot be silenced. Synthetic biology is much broader than that. Another thing that we're interested in is designing bacterial genomes, and so these are the entire either small components of genomes or the entire genomes of microorganisms. And this is was crazy to even suggest that this might be possible even a couple of years ago. But the way that machine learning and generative AI is advancing, it's now possible to really contemplate designing entire genomes that essentially will eventually give us machine learning designed species of microorganism, and then maybe even moving on to eukaryotes and larger organisms eventually. That's possible to contemplate, a little scary to contemplate maybe. Loads of applications of that. So environment, you can then think of designing bacteria that will degrade plastics, will create fuel, rocket fuel, different kinds of food. Another thing that synthetic biology is very involved in is plant engineering. So obviously, GMO is very famous, but you can think about more sophisticated uses of plants with more sophisticated synthetic circuits. A great approach is just purely curiosity driven, and these days, with the internet, with the amount of material available on any subject, and especially with artificial intelligence now that can basically just craft a primer specifically for you. You can tell it exactly what your knowledge level is. I mean, I've started using the research grade ChatGPT Pro, and it gives you really beautifully referenced literature reviews that no longer just go to some dead end or go to a reference that isn't relevant. They go to really high quality reference. So you can basically create I would say today, as of the last couple of months, with this new the quality, because it wasn't previously good enough quality to do this, but the quality of the latest Pro ChatGPT, you can create your own primer on any subject that was tailored specifically to your knowledge level. I would then say, don't just accept what that says. Go follow up the references, read the papers behind what it is, educate yourself on the subject so that you have a good understanding, and then start approaching professors, explaining what you've done and your genuine interest. I would say the number of people that come and approach me that have a genuine, real, just personal interest in a subject and are coming to me with a real, genuine fascination in it and have clearly read a lot and understand a lot already are exceedingly few. So if you do that, you're going to stand out as a young scientist that will catch their attention. And I don't care what age someone is. If they have a real, genuine passion for it and they're into it, I'll try to give them every opportunity that I can to get a jump start in their career. And I'm sure lots of other PIs are the same. Yeah. There's a few different ways, yeah. So I mean, I'll say that my lab, we've had a lot of undergraduate students, summer students and thesis students, and volunteers, so a whole different. So we have anything from 2 or 3 to 10 undergrads working with us at any time doing various research projects. And I actually really like when someone, it's especially attractive to me if someone comes along and they seem like they want to do quite a long stint. Like they'll want to do, say, volunteer for a semester, then do a summer studentship, then do a thesis. And then I can sort of think, okay, this person, we can really put a lot of investment into their training and give them a really interesting project. So I like to give undergrad students pilot projects that I really don't know whether it's going to work or not, but they can be really quite crazy and out there. I don't like to just give them like a little sub-project that's a little sliver of someone else's project. I like to give them something they can really get their teeth into and take ownership of, and just be mentored by the students around them. There's a few different ways to do it. Many people just cold email, and that's fine. I would say anytime you do that, don't send a generic email, because we get so many generic emails that are just clearly not personalized for the target professor. That, you know, those are the ones that are just quickly discarded, because we have to have a way of filtering it. It's really obvious. I would say make sure you understand what the lab does, and write a tailored email. It is a lot of work, but it's worth it, because you're probably going to want to try and get into the best labs, and if you want to get into the best labs, you're going to need to do that. I would say an additional approach, which I would actually say is better than just emailing, is to contact students in the lab and try to get to know, because students are usually pretty open to chatting with other students. So contact graduate students, other people in the lab, have a chat to them, have a coffee with them, talk to them about the lab, and then if they like you and you get on well with them, that student will then talk to the professor about you and say, you know, I've got this student along, they seem pretty good. Do we have any opportunity for summer studentships at the moment? And even if they don't, if with that kind of approach, you may just be able to get in, and with a graduate student or a postdoc advocating for you, and then maybe they'll take you on as a, you know, maybe the postdoc or the graduate student will be willing to mentor you if they like you and get on with you. So that's a good approach. I'd say that's the best approach.
Okay, come with me. I'm a PhD student here in the Garton Lab, and we're going to be doing just a quick walkthrough around. So let's see. So this is the freezer farm where we keep our -80s and our -20 freezers, along with some fridges. These are basically where all of the reagents are kept and all of our samples are kept for long-term storage. And this right here is our liquid nitrogen tank. When you open it, a bunch of like vapor comes up, and it honestly looks pretty cool. It's very cold. All of our cells are kept in here for long-term storage. So we come in here whenever we need to thaw a new cell line. Going into the main lab, we have centrifuges that basically spin our samples. We have a few of those in the lab. We have nanodrops that basically measure DNA and RNA concentrations. And we also have a few members in the lab here. So I'm a fifth-year PhD student, and my project is basically engineering synthetic genes and testing them in human-induced pluripotent stem cells. And I'm finishing up the last bit of my experiments, but there's a few people here that also do like similarly cool and interesting work in synthetic biology. So,
hi everyone. So my name's Aaron. I'm a fifth-year student as well, PhD student in the Garton Lab. Right now, I'm doing maybe not the most glorious of work. I'm dumping out the media that I use to grow some bacterial cells. And the reason why this is really important, even though most of our work involves working with human cells, see that little kind of white thing at the bottom is a pellet of bacteria. So I spun it down really fast in a centrifuge to pull down the bacteria to the bottom of the tube and leave all the media at the top. And I'm going to dump the media out. Eventually, the DNA that's stored in this bacteria, we're going to extract it out and put it into human cells where it's going to have a variety of different functions. But generally speaking, we do a lot of stem cell engineering in this lab. And what this DNA is going to do is express a foreign fluorescent protein from a jellyfish in a human cell. And then we can see it on our microscope, which maybe Danielle will show you later.
That's good.
Yeah, thank you guys.
Spend it after and like.
Yeah, so afterwards I'm going to do a protocol called a mini prep, which is just kind of like a very basic protocol we would do in the lab.
Is that like the silicon membrane, like washing it?
Yeah,
Okay, really nice.
exactly. You've probably done that in your teaching labs. So yeah, I'm going to resuspend this in a buffer and start a mini prep tomorrow. I'm just going to freeze these pellets because I don't have enough time to deal with this today. So, to be honest, but that's the idea, exactly.
Julio, maybe tell us like who you are and yeah, what you're doing, what you're up to.
Yeah, so hi, I'm a research associate in the Garton Lab. I have various projects. I also manage some students. Right now, I'm not doing anything particularly interesting. But later today, I will do qPCR, which is essentially using a PCR machine that has also a spectrophotometer. So that basically can measure how many copies you are making of a DNA sequence over time. And then this will help me to quantify the virus I'm making. And yeah.
Yeah, nice.
But
Cool.
Yes, but it's going to be very long, so yeah.
Yeah, we don't really do qPCRs often, so Julia over here has been doing quite a bit of them recently. So, okay, so basically I can give you a little tour around here. This is my lab bench, and I was doing a lot of PCR purifications here. These are some of my tubes, and I'm going to be doing a Gibson assembly with all of these fragments to essentially build the plasmid that Aaron was talking about to put it into human stem cells and then see and test their functions with these genetic circuits. So what made me want to pursue a PhD? So essentially, I graduated during COVID. Actually, COVID hit in my third year of undergrad. And then I was like kind of like really nervous, to be honest, and maybe alongside a lot of other third year and fourth year students that they didn't have much lab experience. And then more so with COVID, there was even less opportunities. So I was kind of freaking out. And then I was like, okay, I have a lot of extracurriculars that I'm a part of. One thing I know I love is synthetic biology. So I joined my iGEM team here at U of T. I did that for 2 years. And I basically try to get involved with however I could get involved in synthetic biology, since it's not a very recognized undergraduate path. So I did a bunch of extracurriculars. I joined Synthetic Biology Canada. I did iGEM. I reached out to people in synthetic bio to actually know what it is. And I learned a lot about it. Then I contacted my professor, Dr. Garton, to see if there was any available positions in his lab, since it is a synthetic biology lab. And it also combined my passion in regenerative medicine, working with stem cells. So it was just a win-win on both fronts. And this lab was doing just that. And thankfully, our ideas aligned, and I really loved the atmosphere and all of the people that are in the lab along with Dr. Garton. So it was just naturally a good fit. I would say, I didn't really, I applied to a few other places, but this one was the one that stuck out for me because it did combine those two passions of mine. And advice I would give would basically be follow what you like, but follow it very, very in depth. So I did everything I could to learn as much as I could at the time about synthetic biology, and then your passion will basically shine through. So that was essentially how I got here. I got in biomedical engineering in 2021. I started out in the master's program, and in the master's program, you can actually do a transfer to a PhD. So that's what I did after just over a year of the master's. So now you won't graduate with a master's; you'll just graduate straight with the PhD. So that's kind of the path I took. Yeah. Is there anything else? Okay. Cool. Okay, and then I will show you guys. This is our TC room. So this is our biological hoods, and this is where we have to keep everything sterile inside because we do open our tissue cell cultures inside. And since we do work with a lot of bacteria, which you just saw, keeping these two rooms separate is very important. So you have to switch gloves, wear a different lab coat, and everything in here is like as sterile as possible. So I'll just show you one of my plates here. So if you want to come over here on this side, so this is an Echo. It's worth this whole machine probably around $50,000. It has an iPad integrated into it. I can show you some fluorescence of my cells. So let me just focus. So these are some of my cells. These are expressing GFP. So this was the green fluorescent protein from jellyfish that Aaron was talking about. And they also express M-cherry. A little bit dimmer, but you can see that the same cells express both fluorescent proteins. And you can actually see the morphology of the cells here. So these are my stem cells. Can get it focused. So all of these are kind of dead cells, and then these main clusters over here are the stem cells. They are on something called ROCK inhibitor, which makes them really spiky. So that's why they look very, very spiky right now. But yeah, I passaged them yesterday, and then this is how they look with those two like fluorescent markers. So I have a lot of different conditions going right now. So this is just one out of like this 96 well plate. So I have to change media later today. So I will put this back into 37 degrees. But yeah, this is our TC room. Here we warm up media. This is called a bead bath. So this is where I'm warming up some of my media. And then this is the water bath where you can thaw cells. And they're warm as well. It's at 37. Right. So the water bath is, I would say like a little bit more like, it could get contaminated like a bit quicker if you're not on top of it. So the beads you can like autoclave regularly, so nothing's like growing in there. So yeah, so basically like in this area we'll make like the genetic circuits and just cloning. So basically Lego piecing like bits of like DNA together, and then once you have like a complete like set of DNA with all the parts that you want together, you'll take it into TC where we just came from, and then there's a protocol to put that DNA into the cells. So then the gene expresses in the cells.
Was your iGEM project kind of related to your current work?
My iGEM project was not. iGEM, so back when I did iGEM, we were working on an enzyme that degrades plastics.
Now you're doing mammalian cells? Or?
Yeah, now I'm doing mammalian cells. So like there was no mammalian cell work at all in the previous iGEM project that I was a part of, but it just served, like the same techniques are applicable. There was also cloning, and cloning you use in any synthetic biology lab. So there's a lot of just like applicable skills that you learn for both, even though it's not like the exact same. So I would say if you want to get into synbio, like you should learn cloning. There's this really good website. It's called AdGene, and there's a really good cloning book in there. And that's how I honestly started, and I like learned how to clone myself from that book. So if you're interested, you can like check that book out. Or I also did like a synbio kind of course. They're not around anymore, but I was a part of this club called CSberg, and it was a synthetic biology education research group. And some of their videos are actually on YouTube, and they'll teach you cloning, like step by step. Some of the videos are upwards of like an hour long, but if you do want to get into synbio, then it's really good because there's no specific class that actually teaches all of the steps for cloning. So yeah. that's essentially it. If you want a good transferable skill, do cloning.
Awesome.
Yeah.
Thanks.
Yes, I'd say one of my students who came in as a summer student and went on to do a thesis, and now she's actually a graduate student. Her project involves creating human muscle from stem cells and implanting it into animals and eventually we'll put it into humans. And the muscle makes therapeutic proteins which get delivered to the bloodstream and will correct the disease, particular types of disease where a particular protein is missing from the bloodstream. Well, I've yet to meet any PI that I've got to know well that doesn't themselves have imposter syndrome, and I would put myself in that category. And I wouldn't probably hire anyone or be interested in bringing anyone to the lab who doesn't have it, because it's actually really good for you. It gives you a bit of an edge. If you feel like you've got to prove yourself, you're complacent and you think you're great and you don't think you need to work hard, you don't really interest me. People who have a little bit of self-doubt, maybe a little bit of anxiety, they're the kind of people you want. They're the kind of people who do the best science. The kind of people who are really careful, work really hard, are constantly questioning themselves and rethinking how they're doing things, constantly trying to improve themselves and improve their knowledge and expertise. Imposter syndrome isn't a pleasant thing to experience, but it's actually very valuable to you in being a diligent and thoughtful and careful scientist. Obviously, there's a threshold of imposter syndrome, anxiety, self-doubt that is negative. Once you have above a certain threshold, then it's going to impact your work and impact your ability to. I think it helps as a scientist to be a bit delusional. I think it's good to have self-doubt, but at the same time, you've got to believe that your project is going to work, no matter how improbable that is. You have really got to believe that it's eventually going to work. Otherwise, you're just not going to even try. And so, I look for people who have vision and want to do big things and want to try crazy things and take risks. Yeah, so I think for first year student, people just coming into the field, it's very difficult to have a big vision about where you want to go with your science and research, and even more your career. So I strongly advise, and this is often against my own interest because I get really good students and I want them to stay in my lab as long as possible. But to be true to my own conscience, I have to tell them this advice, which is go into as many different labs, into as many different diverse scenarios and experiences as you can get. Experience as many different things and as diverse a range of fields and environments as you can. And that is what will allow you to understand what you like best and what interests you the most. You'll find also that by doing that, you're able to see connections between different fields and different environments that other people aren't seeing, and will maybe give you an edge. That you won't be aware of until maybe 5 or 10 years down the line. You'll suddenly realise, no, that thing I worked on 10 years ago, I could actually apply that in this scenario, and no one else will have thought of that because no one else will have worked in both of those different things. That's a big part of research, is understanding and learning how to deal with failure and things not working. And day-to-day research, most research projects have a very exciting beginning where you're planning and formulating your big idea and your big project plan. Then there's trying it, and a few things work, and it's all great, and everyone's excited. Then you hit that one thing that's going to be your big obstacle, and for some people that's 10 things in a row, and it can be months and years to troubleshoot those problems, and you have to be really creative in thinking of new ways around it. You've got to understand, when is it time to call it a day and go on a different avenue with your research. And so these things are, painful and difficult and very challenging. And it's good to be in an environment where everyone around you has experienced or is experiencing the same thing. And so I think being in a supportive environment, both with the supervisory people that you have, but also your peers, helps to get through those kind of things. Another thing I would say is learn what times and days work for you in terms of when do you have your most clear-minded periods? So is it early in the week, early in the morning? Is it in midnight on a Saturday night? When are those times of the week when you're the most lucid and you're thinking the most clearly? And schedule those times to do your most important work. Like when you've got to write a particularly, an introduction to a paper, for example, or you want to think through a difficult problem, schedule those times to do that. So I would say also be ready to use times when because sometimes you don't have particular times that will happen, but it will just appear out of nowhere. Suddenly you'll feel like, okay, I feel really, really sharp today. Be ready to abandon the thing that you were going to do and jump on the hard thing that you need to do, and use that time to work through that hard problem, and leave the whatever it was that you were going to do that's kind of more mundane. Leave that for a time when you're just feeling regular levels of mental clarity. The last thing I'll say, and this is something I think is quite unique to me, which is something I've discovered, which is because I'm paralyzed, I often find myself, or fairly regularly find myself in situations where I can't use my phone or computer, and I have and I'm on my own for maybe a couple of hours, and I just find myself using that time to think very deeply about problems, about project ideas, about science-related things, and I find that time that's completely uninterrupted by using your phone, using any electronic device, talking to anyone, anything like that, just pure thinking time for many hours on end is incredibly valuable, and I found that that's been incredibly valuable in formulating ideas and solving problems. And it's something that I don't think any of us actually experience at all in life. And I just, by circumstance, have discovered how powerful that is. And I've since come across famous authors, that have said something similar, like they'll go on annual retreats for a month with no phone, no laptop, nothing, just purely thinking time. And that's what gives them the ability to write the books they write. That's what I've read. So I'll just say that just because it's not just me saying it. It's something that I don't think anyone really thinks about prioritizing. Pure thinking time that is uninterrupted by distractions is really valuable. I'd advise people to think about scheduling that for themselves and see what happens. Yeah. I don't think, so the answer to this is that everybody's on a different spectrum in terms of their emotions, and I don't think anyone has the ability to have full mastery over that. You cannot switch your emotions on and off at will, and so you just have to accept that your emotional, I think of it as weather. So your emotions are like the weather. You can't control it. It's coming. It's coming and going like the sun and the rain and the snow, and you have to dress accordingly, and so. I would say, I think just understanding that you are an emotional being, an emotional animal, is the first step in understanding when you have emotions. A second thing that I would say is something that I used to do, is have an activity which is completely different to your career, work and science. Have something else going on in your life. For me, it was up until sort of late undergrad, it was being a climber, and there's nothing. I mean, I'm not obviously the best advertisement for climbing, but that was because I was doing something ridiculously difficult, ridiculously dangerous. But there's plenty of rock climbing you can do that isn't dangerous, really. But something like that, I used to find if I went climbing, it put everything in life into perspective and completely reset my mental state. And it could be something else for you. Something different for other people. It can be anything. Even reading a book can put you in reading a book of some fiction or other can put you in a completely different conscious state and allow you to kind of reset. And that's really important. Also, getting breaks, getting mental breaks from what you're doing, so that you're not just completely focused on one thing for a long time. Your brain actually will produce things that you didn't expect if you do that. sometimes the best ideas and the solutions to problems come when you step away from a problem and think about something else completely. Personally, my advice is to start out, as I said before, by trying to get exposure to as many different things as you can. Once you've found something that that tickles your interest and you find engages you, follow that interest. And I would say continually be updating what you're interested in. I would say when you're setting out on your career, think of the thing that you think will be the most interesting, the most cool thing you could possibly work on, and go and try and do it. And you'll find that you probably can. Don't just think, don't have self-doubt, oh, I probably couldn't do that. Just go and try it and do it. And you'll find that if you really want to do it and you're excited about it and you're interested in it, not that many people are genuinely enthusiastic and excited and interested in any one particular thing. And so if you are that, you're going to stand out among the other people. And so if you follow your nose and follow what you're genuinely interested in and what excites you, you are going to stand out and you are going to do a good job of it, and you're going to have an interesting and enjoyable life. The money, the finances, the success, all of that will kind of naturally come if you're doing something that you're genuinely excited and interested in. I know it's kind of a bit cliche. It's probably very, very kind of mainstream advice, but that has been my experience, and I think it's genuinely true that you're in for an interesting and enjoyable life if you are continually thinking, what is the most interesting thing that I could be working on? Okay, I'm going to work toward that always, and that changes through your life. And it's still changing for me. I'm continually correcting my trajectory and direction according to what is the most interesting thing I can think of at any given time.